Beyond birthdays, how biological age may better predict dementia risk than calendar. Age time is an unforgiving constant in human existence. We measure our lives in years, marking birthdays as milestones that supposedly indicate our progression through life. However, medical science increasingly recognizes that the number of candles on your birthday cake may be a surprisingly imprecise measure of how your body is actually aging. This disconnect between chronological age, the years you've been alive, and biological age, how your body is functioning relative to established norms, has profound implications for understanding, predicting, and potentially preventing age-related diseases, including dementia. In the medical community, age stands as perhaps the most reliable predictor for numerous chronic conditions. As Dr. F. Perry Wilson from Yale School of Medicine notes, there's even a running joke among medical students that when asked about the #1 risk factor for virtually any disease, age is the safest answer. From cancer and cardiovascular disease to kidney dysfunction and cognitive decline, the passage of time correlates strongly with increasing risk. Yet this observation raises a fundamental question. Why should the number of times we've orbited the sun dictate our susceptibility to disease? This question has driven researchers to develop the concept of biological age, a measure that attempts to quantify aging based on physiological parameters rather than calendar years. The appeal is twofold. Biological age potentially offers a more accurate assessment of disease risk, while also representing something potentially modifiable, unlike the relentless march of chronological time, measuring what truly matters: the science of biological age. The measurement of biological age remains a developing field with various competing methodologies. Some researchers favor DNA methylation patterns, often referred to as epigenetic clocks, which measure chemical modifications to DNA that accumulate with age. Others employ phenotypic clocks that incorporate more readily available measurements like blood pressure, lipid profiles, and inflammatory markers. A recent study published in the journal Neurology examined two well-validated biological age calculation methods: PhenoAge and the Clemmerer-Dubel biological age measure. PhenoAge integrates information from 9 blood biomarkers, including albumin, creatinine, glucose, C-reactive protein, and white blood cell count. The Clemmerer-Dubel method incorporates these markers, along with additional parameters to create a comprehensive aging assessment. Interestingly, even these two established methods showed relatively weak correlation, coefficient of 0.29, highlighting one of the challenges in this field. Different biological clocks may be measuring distinct aspects of the aging process. This diversity of measurement approaches raises important questions about standardization and interpretation of biological age assessments. The UK Biobank study, linking biological age to dementia. Risk. The Neurology study leveraged data from the UK Biobank, an ambitious research initiative containing medical information, genetic data, and biomarker measurements from over 500,000 UK residents. Researchers identified more than 280,000 individuals with sufficient data to calculate biological age, who also had available genetic information and no dementia diagnosis when entering the study. Over a follow-up period exceeding 10 years, 4,770 participants received new dementia diagnoses. The analysis revealed that older biological age, regardless of which clock was used, significantly increased dementia risk. However, the most telling measure wasn't biological age in isolation, but rather the gap between biological and chronological age. Essentially, how much faster or slower someone was aging compared to calendar expectations. The findings were striking. For each year that biological age exceeded chronological age, dementia risk increased by approximately 1%. Put practically, someone with a calendar age of 65 but a biological age of 75 would face roughly a 10% higher dementia risk than a peer whose biological and chronological ages were aligned. This effect was consistent across both biological age measurement techniques. Perhaps most remarkably, the impact of accelerated biological aging was dramatically amplified among carriers of the ApoE4 gene, the primary genetic risk factor for Alzheimer's disease. Among ApoE4 carriers, those in the highest quartile of biological chronological age discrepancy, the 25% of people with the greatest difference, demonstrated a 4-fold increased dementia risk compared to those with the smallest age gaps. This suggests a potentially dangerous synergy between genetic predisposition and accelerated biological aging that could substantially elevate dementia vulnerability. Beyond correlation, understanding mechanisms. While establishing a connection between accelerated biological aging and dementia risk represents an important advance, understanding the underlying mechanisms presents a deeper challenge. The researchers hypothesized that brain volume might serve as a mediator in this relationship. Essentially, that faster biological aging might accelerate brain shrinkage, ultimately leading to cognitive decline and dementia. MRI studies partially supported this theory, suggesting that about 13% of the link between biological age and dementia could indeed be mediated through brain volume. However, this leaves the majority of the relationship unexplained, pointing to other potential pathways through which accelerated aging might influence cognitive health. These could include vascular changes, inflammatory processes, impaired cellular repair mechanisms, or other factors not yet identified. This complexity highlights both the challenge and opportunity in biological age research. While we can observe the statistical relationship between biological aging and dementia, fully understanding the causal chains involved requires further investigation. Each pathway identified represents a potential intervention target for reducing dementia risk. The promise of malleability: can we change our biological age? Unlike chronological age, which advances inexorably regardless of lifestyle or medical intervention, biological age appears modifiable. This represents perhaps the most hopeful aspect of this research direction. Various factors may influence biological aging rate, including diet, physical activity, sleep quality, stress management, and environmental exposures. Even small changes in individual biomarkers can meaningfully impact calculated biological age. For instance, reducing C-reactive protein, an inflammation marker, by 0.5 point could potentially lower biological age by approximately 1 year using the PhenoAge calculator. Other interventions that might favorably influence biological age include improved glucose control, blood pressure management, weight optimization, and reduction of oxidative stress. While we don't yet have definitive evidence that deliberately reducing biological age will directly decrease dementia risk, correlation doesn't necessarily equal causation. The prospect is tantalizing. If the connection proves causal, focusing interventions on reducing biological aging rates could potentially represent a powerful approach to dementia prevention, particularly for high-risk individuals like ApoE4 carriers. Practical implications: from research to application. The emerging understanding of biological age has several important practical implications. First, it suggests that risk stratification for age-related diseases like dementia might be significantly improved by incorporating biological age assessments alongside traditional risk factors. This could help identify high-risk individuals who might benefit from more intensive monitoring or intervention, even if their chronological age doesn't suggest elevated concern. Second, the concept of biological age provides a potentially powerful motivational framework for preventive health behaviors. Rather than abstract advice about healthy living, patients might respond more strongly to information about how specific lifestyle changes could reduce their biological age and potentially lower disease risk. Framing health interventions in terms of reversing aging could prove more compelling than traditional prevention messaging. Third, biological age assessment could help evaluate the efficacy of interventions aimed at reducing age-related disease risk. Rather than waiting years or decades to observe clinical outcomes, changes in biological age could potentially serve as intermediate markers of intervention success. This could accelerate research into preventive strategies for conditions like dementia, where traditional outcome studies require extremely long follow-up periods. Finally, biological age research highlights the interconnected nature of age-related diseases. The same accelerated aging processes that increase dementia risk likely also elevate risk for cardiovascular disease, cancer, and other chronic conditions. This suggests potential synergies in prevention strategies. Interventions that slow biological aging might simultaneously reduce risk across multiple disease categories. Limitations and future directions. Despite its promise, research on biological age and dementia faces several important limitations. The lack of standardization in biological age measurement creates challenges for comparison across studies and clinical application. Different biological clocks may capture distinct aspects of aging, complicating interpretation. Even well-validated measures like those used in the neurology study showed only modest correlation with each other. Additionally, most current research on biological age and dementia, including the UK Biobank study, is observational rather than interventional. While these studies can identify associations, they cannot definitively establish causality or demonstrate that deliberately reducing biological age will prevent dementia. Prospective intervention studies targeting biological age reduction with dementia as an outcome are needed, but would require substantial time and resources. The field also faces challenges in translating biological age concepts into clinical practice. Questions remain about how frequently biological age should be assessed, which measurement approach is most appropriate for different populations, and how results should guide clinical decision-making. The cost and accessibility of some biological age assessments, particularly more sophisticated measures like epigenetic clocks, may limit widespread implementation. Looking forward, several research directions appear particularly promising. Further investigation of the mechanisms linking accelerated biological aging to dementia could identify new intervention targets. Comparative studies of different biological age measurement approaches could help establish which methods best predict specific outcomes like dementia. Intervention studies aimed at reducing biological age could help clarify whether this approach meaningfully impacts dementia risk. Finally, research on how biological age assessment might be incorporated into clinical risk prediction models could help translate these concepts into practical applications. Conclusion: a new paradigm for understanding aging and dementia. The emerging research on biological age represents a potential paradigm shift in how we conceptualize aging and age-related diseases like dementia. Rather than viewing aging as a uniform, chronology-driven process, this approach recognizes aging as heterogeneous, multifaceted, and potentially modifiable. It suggests that how we age may matter more than how long we've been alive when predicting disease risk. For dementia, a condition that continues to challenge medical science with limited treatment options, this perspective offers new hope. If accelerated biological aging indeed contributes causally to dementia risk, interventions targeting aging processes could complement traditional approaches focused on specific pathologies like amyloid plaques or tau tangles. Perhaps most importantly, biological age research emphasizes that many aspects of aging that influence disease risk lie within our control. While we cannot stop the calendar, we may be able to influence how our bodies experience the passage of time. This empowering message suggests that through appropriate lifestyle choices and medical interventions, we might slow the aging processes that contribute to dementia and other age-related conditions. As science continues to unravel the complex relationships between biological aging and disease, the distinction between how old we are and how old our bodies believe us to be may prove increasingly crucial to health outcomes. Beyond birthdays, the true measure of our age, and its implications for cognitive health, may lie in the cumulative effects of genetics, environment, and lifestyle on our biology.